Really? Good afternoon. Welcome back. Welcome back. Welcome back. So now, at this point in time in the semester, we start our sprint towards the finish. I find that every year, the first part of the semester up to spring break is kind of slow. You kind of like you're trudging through, and then you get to spring break, and all of a sudden, it's all this stuff is ongoing. We got meetings and preparing for graduation and everything, all this stuff happening at once over the last 6 weeks of the semester coming up, and boom, all of a sudden it's May, and you guys are like, oh, I want to leave. All right? So this is a very much a sprint, okay? The last leg sprint of the semester of the academic year. So we have still quite a bit to finish, to get through. We still have to go through the respiratory system, the digestive system, the reproductive system, the urinary system. So we have multiple systems to get done. But before we do all that, let's finish up on the immune system. I'm going to try to get the immune system done by Wednesday, if not the first part of Friday. Okay, and move on to the respiratory system. But to review, because I know that you haven't really done much over the break, to remember where we're at. We had finished the first part of the immune system, which was on your exam, right, which covered innate immunity. All right, and this graphic shows you what innate immunity includes, right? What I call the first and second line of defenses, right, are non-specific defenses. First line of defense is to keep things out, the skin, the mucous membrane, secretions of the skin that prevent things from entering into the deeper cells. But in case they do get past that first line of defense, we have another non-specific type of defense, which includes phagocytic cells such as macrophages, natural killer cells, antimicrobial proteins such as complement, all right, and lysozyme, and then the inflammatory response is included in this, and I actually said that we could include fever as well as another line of defense, or at least a hallmark, an indication of something that's wrong. Okay? But what of all those have in common is that they are non-specific body defenses. They do not care whether it's a bacillus bacteria or a streptococcus bacteria or a diplococcus bacteria or whatever, or an amoeba that gets past that. They don't care what it is. They will deal with it the same way. Okay? It is not a specific defense against a specific pathogen. All right? All of that is non-specific, innate defenses that you are born with from the beginning. Now we move on to the next part, the adaptive immunity. All right? Adaptive immunity includes your third line of defense, lymphocytes, right? And what they do, there's two types of lymphocytes, T cells and B cells. We'll talk about them today. B cells produce antibodies, which will react to certain antigens. And we'll talk about antigens today as well. And then distinct, specific macrophages. So macrophages here, right, are also included in this. And so second line of defense kind of can transfer over to the third line. When a macrophage engulfs a bacteria, digests it, and then uses part of that bacteria, that digested material, to initiate another response from B cells. All right? And then breaks, or what we call, presents that antigen, that piece of cell wall, protein, whatever, that has been digested from that bacteria, and then presents it to a lymphocyte so it can produce antibodies or have another cellular response. We're going to talk about both of them. So really we're at the adaptive specific immune responses. Okay? So adaptive immunity involves specific lymphocyte responses to specific antigens. Okay, so this is really the basics or the basis for vaccinations, all right, or why you have vaccinations, all right? So what starts in the adaptive immunity is some type of signal, something that is recognized, right? And that recognition, that ID card, is what we call an antigen. Okay? An antigen, we'll go over what it defined as in a second, but it will be the signaling point, the starting point, the initiation of this adaptive immunity. Okay? If there's no antigen recognition, there's no signal to start. There has to be some type of antigen recognition. Okay? And then that starts the immune response. Okay? And that consists of what lymphocytes do and what they produce within that and the responses that they elicit in the body. Okay? This is different. How is adaptive immunity different from innate immunity? It's developed and it's specific. Okay? And it takes a little bit more time. All right? It takes a little bit more time. So you probably walked up the stairs and down the hall here, and you, as each person, probably have been exposed to a number of pathogens walking up the stairs, many of which you won't even know that you've been exposed to. Your immune system doesn't know you're exposed to them because immediately your mucous membranes trap those particles and they're disrupted, they're immobilized, neutralized, whatever you want to call it, all right? They don't elicit an immune response. Because now, but say one, you touch your nose and now you have a viral, a cold virus. Now that moves past that second line of defense into the third, and then you could develop an immune response over the next couple days to that cold virus. All right, so it takes a little bit more time because of the way this works. It takes a couple days to develop. All right. There are two branches of adaptive immunity we're going to talk about. Okay? One is cell-mediated. What do you think cell-mediated means?
Mediated by cells.
All right. It is the function of the immune system is mediated by cells. Okay? And what the cells do. The other one is humoral immunity. If one is mediated by cells, the other one is mediated by something that's not cells. It's actually something that's produced by cells. Humoral immunity is mediated by antibodies. Okay? So, antibodies. So, effective against antigen outside of cells and does not require, right? But this is the big one. Produces antibodies, which will do something. And we'll go through the reactions that occur with antibodies in a little bit. Okay? But the cell-mediated one is mediated by T lymphocytes, T cells. All right? And it's effective against antigens within cells. All right? While the others are not, humoral is effective against antigens outside of cells. And we'll talk about what that means in a little bit. All right? And there's a couple different types of T cells we'll talk about, cytotoxic versus helper. All right? And then what it does, it actually induces cells to die. Okay? So there's actually a little bit of difference in here. Right? One big difference, antibodies versus cells. The second big difference, the type of cells that are used, B cells for humoral, T cells for cell-mediated, and where the antigens are that are targeted. Okay? Because the humoral immunity is mediated by antibodies, the antigens have to be outside of cells. Okay? And because the T cells can actually work against antigens inside of cells, all right? Or that's one of the differences. Okay, questions on that? T cells, B cells, and the two branches of adaptive immunity. All right, so let's talk about antigens. Antigens. Now, not all antigens are pathogens, all right? But let's talk about an antigen. An antigen is anything that can kind of start an immune response, okay? So all your cells, and all cells in general, have these parts of their cell membrane and their outside ID cards that live on the outside of cells that are recognized as either you or not you. Self versus non-self. So I put up there pathogens, and I put in parentheses non-self materials. Okay? Non-self materials. And I said materials, you could say substances or whatever, because it's not necessarily just proteins. It's not just necessarily just carbohydrates. It could be anything. Could be an antigen. Okay? As long as it kind of induces an immune response. But the point of it is self versus non-self. Your body, your immune system uses these ID cards to determine what is you and what's safe versus what's not you. And they want to attack, or at least regulate, what's not you in your body, because that causes problems. All right? So an antigen that binds a T lymphocyte or an antibody. We'll talk about antibody-antigen relationships in a little bit. Usually, an antigen is a protein or a large polysaccharide. Examples, the protein capsid of a virus. All right, so if we want to draw a little virus out here, a little bacteriophage virus, right? Genetic material. Part of the protein that's out here, right? Part of the tail proteins, whatever, could be considered an antigen if it is broken up and is recognized by the immune cells. Cell wall of fungi or bacteria, right? So bacteria have a little cell wall. There's a circular plasmid, but on the outside, they have another outer cell wall. Part of that could be used, broken down, and used as an antigen. Be recognized as, right, as non-self. Bacterial toxins, abnormal proteins, or even tumor antigens. Right? During tumor progression, cells behave differently, and they can develop different ID tags on the outside of cells that are mark them as being tumor cells, partially as a response for the immune system to remove them from the body. Okay? So the point of an antigen is an ID card for every cell of your body. And when did we talk about antigens previously? What are those? What's this thing? Let's put it this way. Now what are those? Taylor. Antigens that determine your blood type. All right? A-type antigen, B-type antigens, or you don't have them. All right? So you've already talked about antibodies and antigens. All right? Antigens identify what type of cell is there and who it belongs to. Okay? The antigens that say that is what's you is you, it's a human cell or it's a mouse cell or something like that, they're on every one of your cells, and your immune system has receptors for those. They'll run into it, and they will identify it. Oh, that's you. Okay, you're fine. You can come into Clubhouse Cafe. Welcome to the clubhouse. Just saying. If that's where you would like to go or whatnot. You know? All right? So, foreign antigens versus self. All right? So, self versus non-self. Typically, you don't bind, or you recognize self-antigens and it doesn't elicit an immune response, so it moves on. Sometimes that doesn't actually happen. All right? You end up with what's called an autoimmune disorder. All right? Where your immune system is attacking your proteins or carbohydrates or cells of your own body. rheumatoid arthritis, multiple sclerosis, right? Lupus are all examples of autoimmune disorders where the immune system is working to fight off or responding to a normal antigen that is normally present in your body, right? But now, for whatever reason, it is eliciting an immune response and causing problems, all right? Now, an antigen can be lots of different things, and we can even break it into different parts. So I said an antigen could be a part of a protein capsid. It could be a capsid protein from a virus. It could be part of the cell wall of a bacteria. All right? And we can break it into further parts by using the term what we call an epitope. All right? An epitope is the specific region that is of that antigen. Okay? That's recognized by the immune system. All right? So when if you're doing research and you want to look at the antibody you want to use the antibody to see if, I don't know, some protein is present in cells. You go on internet, you look it up, you find the antibody, and there's like these lists of 30 different antibodies for the estrogen receptor. All right, human estrogen receptor. Well, they're all the same human the antigen that was used. They used the estrogen receptor. But if they use different parts of the receptor, they can actually elicit different responses, right, from the immune system of the animal they were using. So it actually targets different areas. Think about it like this. How many of you got a flu vaccine this year? How many of you got a flu vaccine the year before? Why'd you get them every couple of years? It's a different strain, right? Do they use the same vaccine? No, they have to change it, right? Still the flu vaccine, it's still the flu virus. Well, why do they need to change it? Because the number one, because the virus evolves or mutates. But second, they actually can use different epitopes. Okay? So if you say the flu virus is this complex virus, and if we use one part of the protein, capsid protein, we can use a different part of the capsid protein to make a different vaccine. And every year they develop the vaccines and the multiple vaccines, and they go through and find which one is most effective for the particular virus they're looking at. Right? So the epitope can actually, you can have multiple epitopes for one particular antigen. Let's say the flu virus is one antigen, the whole capsid, but there could be multiple parts, multiple epitopes of that particular flu virus antigen. All right? So we can have what's called multiple determinants and multiple epitopes. All right? So here, what they're showing, one particular antigen. Antibodies that have been produced by B cells that are responsive to several different epitopes on the same antigen. Okay? Some other distinctions. Immunogens. All right, is an antigen that induces an immune response. Okay, and then immunogenicity, ability to trigger a response. Okay, so if something's immunogenic, it will cause an immune response. And we want that to happen. So if we're making antibodies, let's say we're making antibodies for research purposes, right? We want the animals that we're using to produce those antibodies to have an immune response. So we inject them with, let's say, a killed version of your chickenpox virus, right? And the chickenpox virus goes in, and now we want them to have an immune response, but it's a killed version, so they may or may not. So we inject them with another protein or carbohydrate that elicits an immune response, and then can make it more robust. Okay? And then we have haptens. A little bit different. Small foreign molecules that induce an immune response when attached to a carrier molecule in a host. When attached to a carrier molecule. So by itself, it may or may not be immunogenic, but sometimes it attached to a carrier molecule, another we call adaptive molecule that allows them to elicit the immune response. Toxin and poison ivy is an example, and then account for hypersensitivity reactions. Okay, so anyone in here allergic to penicillin? It's not that you're allergic to the penicillin, it's that the penicillin binds to something else inside you, and then that elicits the immune response and causes problems. Yes, you are allergic to penicillin, you're going to have problems, but go from there. Right? Pollen, right? In about 2.5 weeks, I'm going to hate springtime because I'll be all watery-eyed and stuffy for another 2 weeks. Start sneezing, especially because that pine grove over there, I'm allergic to pine pollen. Oh, it's gonna suck. So bad, gonna suck. Well, all right, so we went over some immune disorders, all right, and what they are: rheumatic heart disease, type 1 diabetes. What's type 1 diabetes? What cells are targeted by type 1 diabetes?
Insulin producing cells.
Which are?
Pancreatic.
The pancreatic, right? The islets of Langerhans, beta cells. Okay? The beta cells within the pancreas are targeted and killed off by one's own immune system. So now they cannot produce insulin, all right? Or insulin is blocked by antibodies to insulin and then can cause problems. Multiple sclerosis. Those of you how many of you did your autoimmune disorder presentation on multiple sclerosis? Kyla, what is the target of multiple sclerosis? She's the only one in here. What's the target of multiple sclerosis?
The central nervous system.
Particularly what?
The
The neurons, right? The myelin sheath of neurons, okay? And so all of these affect different body structures. The immune system being a link between all systems, can affect any, right, if you have an autoimmune disorder. So let's talk about the general structure of lymphocytes. 2 types of lymphocytes, T cells and B cells, right? They differ based upon the antigens they have on their cell surface, okay? So T cells and B cells have unique receptor complexes. They have different proteins on the cell membrane. You have a T cell receptor or a B cell receptor, okay? TCR or BCR. They're specific to the type of cell they're there. So a B cell is not going to have a T cell receptor because it's not a T cell. What are they called? All right? So here's a helper T cell. Here's a cytotoxic T cell. And then you have a B lymphocyte, the cells of humoral immunity. All right? T cells can be further categorized into the type of antigens they have on their cell surface. All right, so not only does it have the T cell receptor in green, which is not present on the B cell, right here. Where is that? Right here. That was weird. I have 2 pointers? No, 1. Okay. T cell receptor, but they also you can be further categorized whether it has a CD4 or CD8. What's CD4 and what's CD8? Those are 2 T cell antigens. Okay? CD4 positive cells are helper T cells. CD8 positive cells are cytotoxic T lymphocytes. Different antigens, different types of cells, different types of cellular function. Okay? And then you have your B cells, which have a different type of receptor. They have a B cell receptor instead of a T cell receptor. Okay? In this case, your B lymphocytes, because of their receptor that's out there, what is it? This receptor actually looks quite similar to the structure of antibodies that are produced. They can make direct contact with antigens and clump together. Okay? But T lymphocytes, they are not activated directly by the antigen. Okay? They get activated when another cell presents that antigen to it. Okay? So another cell has to incorporate and meet with the antigen, interact with it, and then do something and then activate the T cell secondarily. Okay? But the B cells, because of their structure and their B cell receptors, they can run right directly into antigens and start. Okay, so helper T cells are CD4 positive. Now, we talked about the fact that there's crossover between innate defenses, nonspecific defenses, and specific defenses. And this is where it happens. Okay? So your helper T cells, they assist in cell-mediated humoral and innate immunity. Okay? So cell-mediated is T cells. Humoral is what? Is mediated by what? John? Antibodies. Okay? So, and antibodies are produced by what type of cells? B cells. Okay? And then also innate immunity, because they can activate these natural killer cells and macrophages to phagocytose any particular antigens. Okay? And then your cytotoxic T cells are CD8 positive cells, and they release chemicals that destroy other cells. Okay? Now there are some other types of T cells, including memory and regulatory T cells, but they have different functions, and they don't necessarily act they have different steps in the pathway. Right? They're not going to elicit immune responses. They're going to help other cells to start an immune response. Okay? So, I talked a little bit about antigen presentation. I've mentioned it a couple times. T cells they do not directly run into and meet up with an antigen. Right? They can't directly bind an antigen. So something has to happen. All right? A macrophage, for instance, will phagocytose a bacteria, digest it, and then utilize some of the broken proteins and carbohydrates as an antigen and present that to a T cell to activate the cell-mediated immunity. Okay? So there's two types of cells that produce and present antigens. All right? The cell antigen presentation, the cell displays the antigen on the plasma membrane and activates the T cell. All right? Chapanga. That's pretty much what's going on. Okay? Yeah. All right, Simba. Okay, right, but it recognizes, the T cells recognize the antigen and then can start a response based upon it. Okay? Presentation. Macrophages do this. Other antigen-presenting cells. All right? Immune cells. All right? Dendritic cells, macrophages. Even B lymphocytes can do that and present the antigen. And all nucleated cells of the body. All right? So something gets into an epithelial cell, it can start that as well. Okay? Questions on antigen presentation? What this requires is that the antigen has to attach to what we call an MHC molecule, a major histocompatibility complex. MHC is a group of transmembrane proteins that are found, some of them are found on all your nucleated cells, right? And the MHC2 is found on antigen presenting cells. So major histocompatibility complex, MHC proteins, are found on all your cells. Okay? And some, a particular type, are also found on antigen presenting cells. So in order for an antigen to be presented, it has to attach to this protein and then get moved by the cell onto the plasma membrane and kind of stuck out there. Little hand waving out there. Hey, hey. All right, how you doing? Antigen here. Okay? Without that, it can't be activated. Right? An antigen itself is not going to be floating around outside of a cell and run into a T cell and start an immune response. Okay, that's not how it could run into a B cell and start an immune response, but not the cell-mediated part. Okay, your MHC proteins are glycoproteins. What do I mean by a glycoprotein? Alexa, what's a glycoprotein? Close. What does glyco mean? What does the suffix glyco mean? The prefix glyco means what? Sugar. Okay? So it's a carbohydrate that is attached to a protein. All right? So it's a big protein molecule, part of the plasma membrane that has a sugar oligosaccharide, meaning a group of sugars on the outside. Okay? And it has genetically determined structure that's unique to you. Why is it important that it's unique to you? It's Taylor Piro, not the other Taylor that sits a couple seats down. Why? Why is that important? Because Colin doesn't want to sit in the back. He wants to sit up here in front.
We'll do it next class.
No, thank you.
Okay.
Good. Distance is good. Distance, good. Little, whatever gets you through the day. Okay? So why is it that important though? Because if it's you this allows you to recognize self versus non-self. So genetically, we have to have a distinction between two individuals. Why is this important and where is it used? How many of you have ever been tested for bone marrow donors? Why do you have to be tested for donations?
Make sure you're a match.
What are you a match for? These compatibility products. Okay? So genetically, what is the highest probability of you finding a match for an organ donor or a bone marrow donor? What's the highest probability? Are you just going to pick a hey buddy off the street, or where are you most probably going to find a match?
In your family?
In your family, right? Close family relationship. Usually, you have a higher amount of genetic material in common. So your closest match is probably going to be someone in your family. If you're an identical twin, what is it? Hey, no, you're not.
What?
Huh? What?
No,
No,
you're not.
no, you're not. Okay?
I'm
He
special.
just told you you're not.
I'm special.
He told you.
I'm going to have to separate them too. Okay. So identical twins are genetically identical, right?
It's a fingerprints.
Okay. That's not genetic, so don't worry about it. So I said genetically identical, okay, for the most part. So they are close. They are the best matches, right? Their MHC complexes should be genetically either the same or very, very close, depending upon any mutations that have occurred. Okay? So these MHC class 1 molecules, they display fragments of proteins, right, that have been brought in. All right? But this is the important part right here. If fragments are from endogenous proteins. The immune system recognizes them as self. So your cells are producing proteins that are produced. They put them on the outside. They're you. They're recognized as self. But if it's from something that's come in from the outside, an infectious agent, a part of a virus, part of a bacteria, part of a fungus, something, part of a cell that's from not you, recognized as non-self, and that communicates to the cytotoxic T cells to kill this cell. It's a flag for destruction. Okay? So this is how this works. They're continuously synthesized off the rough endoplasmic reticulum. Start here. All right, here's a self-antigen. It leaves, goes to the transport vesicle, goes through the Golgi apparatus, eventually gets to the outside, gets to the plasma membrane, and is recognized as self. Yay, happy, you, good. Okay? This happens on all your cells. So your immune system can continuously monitor what's you, what's not you. Okay? But now, when you end up with a foreign antigen, the same process occurs. That foreign antigen is incorporated through endocytosis or gets in the cell somehow, goes through the rough endoplasmic reticulum, gets reprocessed, and then pushed, attached to an MHC molecule through the Golgi apparatus, again gets to the plasma membrane, and now that foreign antigen is expressed on the cell surface. Cytotoxic T cell will recognize that foreign antigen and kill this cell. Okay? Because this particular cell has been infected by a virus that we want to get rid of. This cell will be killed off, undergo apoptosis, right, and undergo lots of different stuff. So let me ask you, how many of you have had a cold within the last year? And if all your hands don't go up, you're out of your tree. Okay? That's a rhinovirus. Virus comes in, affects the cells of your upper respiratory system, your nasal passages, okay? And those epithelial cells start this process, and they produce this, okay? So what do you end up with? All that phlegm and stuff that's happening, and the mucus that's produced is an immune response. It's not that you have a cold. Yes, you have a cold, but the virus is growing and passing from one epithelial cell to another, and your immune system is working to prevent that, and it's killing off the cells, okay? That phlegm and stuff is dead and disrupted mucus cells, it's apoptotic cells, it's white blood cells that are there, lymphocytes that are there, antibodies that are there that are trying to get rid of this virus. Okay? And that's what has occurred. The virus you've inhaled it, right, or gotten it into your epithelial cells in some point. It's got into the cells, been processed, and now the MHC class has presented it, and now the immune response has occurred. While that's ongoing, the viral particle has also hijacked the cell, has reproduced and released more viruses, right, as that cell dies. Right, so there's this ongoing war about trying to get rid of the cells that are infected. Okay? MHC2 molecules are also glycoproteins. All right. Again, exogenous antigens brought into the cell through endocytosis. All right, so in this case, in the first case, we had the MHC1 that came in. All right, this MHC2 is not necessarily their self versus non-self. Okay? And so now phagocytosis occurs, all right, gets that bacteria is broken down, that digested proteins are reprocessed, broken up, attached to an MHC molecule, and then put back on the outside of the cell. So now that macrophage becomes an antigen presenting cell and presents it to a cytotoxic T cell, right, helper T cells to start an immune response. Okay, so this is what's called the endogenous pathway. All right, comes in, secretory vesicle, phagosome, broken down, phagolysosome, and then the secretory vesicle that's here, what am I doing? Is here, transport to the MHC2 proteins, and eventually goes right out there. All right, so you have this part, this normal MHC2 class proteins are going through, and sometime then there's fusion with the vesicle that had the phagolysosome, which has broken up the bacteria, and now parts of that bacteria are presented on the outside that can elicit responses from the cytotoxic T cells. Which one of these, MHC class 1 or MHC class 2, do you think happen more?
Class 1.
So class 1 is produced probably more, right? Which one do you think happens, or does one or the other pathway occur in a particular immune response? Or do they both occur simultaneously? Does one pathway of antigen presentation happen if you run into a flu virus, and then the other pathway happen if you run into a bacteria? Or do they kind of happen together? Sebastian? They happen at the same time. All right, so what's the benefit of them happening at the same time? Is there a benefit? Do you agree there's a benefit that of these two pathways happening simultaneously? Okay, so what? Where's the advantage? Crickets. Okay. So let's look at this picture. Okay. So we have a pathogen that's come in here, and now if you look up here, what color is this particular foreign antigen? It's green, right? But there's other parts of this. Let's say that we had an antigen out here that this is epitope A, this is protein B and C. So now what happens if I do this? I could have A, B, and C as particular antigens, particular epitopes to the same bacteria, right? So now that's one way of doing it. What if the other way of where things are brought in and goes through an endogenous protein is attached to this MHC1 protein that we saw the first picture, and now I had proteins D, E, and F that are presented on that particular cell. So now I have two separate cells that are presenting three separate antigens to the same pathogen. What does that do for your immune response?
It's more likely to be caught.
So it's more likely. Kayla's like, I know what you're trying to say, and I know it's in my head, but more likely to be caught. Yes, it's more likely to be caught, right? It's more likely that your immune system is going to have a bigger response to it, right? So not only is it going to recognize antigen A, but B, C, D, E, and F all at the same time. So if any of those antigens are presented on the cell, that means that cell is going to be dead, going to be killed off. It's a way of augmenting your response. You have a larger response, okay, to these antigens. Okay? And then you can actually here's the interaction that occurs, right? So you have your nucleated cell, a nice epithelial cell in your nose that has incorporated a virus and now produces that. Let's say at MHC class 1 or class 2, interacts, that presented antigen interacts with the T cell receptor, the T cell receptor protein. Okay? And your CD4, now that takes that and recognizes that antigen, and now that activated, and now this cell would be killed off by the T lymphocyte. All right? Or it could take that antigen and then induce the production of antibodies from a B cell. Okay? So these antigens that are presented end up binding with the T cell receptor, okay, on your helper T cells. All right, so let's talk about your MHC molecules in terms of organ donations. All right, so we know that you can transfer organs from one person to another, but you have to be tested for a match. All right, and they're based upon this. So you're tested not only for blood type, okay, so because that's your first indication of whether or not you're going to be a likely match, but you also have to be tested for MHC compatibility. Okay? There are no two individuals with exactly same MHC molecules. Right? So even identical twins are not exactly the same because of mutations that have occurred over time. But they are your most likely family members, close family members are your most likely matches. Okay? So when you get an organ transplantation, all right? You get an organ transplant and it's inside you, what do you have to do for the rest of your life? Taylor.
Take anti-rejection.
You have to take immunosuppressants, okay? Anti-rejection medication. You have to suppress your immune system so that it does not go and recognize that that's not you and start to kill it off, okay? And then you're immunocompromised for the rest of your life, and you can go from there, all right? So it prevents rejection, right? I'd rather deal with the colds and everything else I have to deal with in order to keep my functioning kidneys. John?
So if you had the same human blood group as the individual, it would still target it as foreign?
Yes, because you don't have exactly the same MHC molecules. Okay, you get as close as you can, all right, so that it reduces the chances of rejection, and then you end up with immunosuppressants to allow that to go for right. There are some organs that are transplanted without that. The cornea, you can have the cornea that is transplanted, right, from one person to another without any rejection, right? Some cadaver. Any of you ever have an ACL tear or replacement? You take cadaver ACL and ligament and put it in your knee. Go from there. All right, so we'll pick up on this on Wednesday. And welcome back.